And one. All right. So, I told you. Wow, what is happening? Okay. All right, so I told you we are going to take this day today to get ahead if it was possible, because next week we only have Monday and Wednesday, and the following week we only have Wednesday and Friday. And so, yeah, next week, Engage is Thursday, Friday, Saturday, Sunday as well. And so there are no classes Thursday, Friday next week, nor are there any night classes on Wednesday of next week. And then Monday is reading day, and so there are no morning and or afternoon classes. There are evening classes on Monday. Anyways, all right, so here we go. Chapter 27. Introduction to animals. First framing question is one we've dealt with before, but outside of animals. Is there a relationship between complexity and diversity? But now we're going to apply it specifically to animals. How have Hox genes contributed to the diversity of animals? And the third one we'll probably get to today, explain humans in terms of animal classification schemes. All right, first one. What is the relationship between developmental complexity and animal function and diversity? We've explored this before, and what did we determine is the relationship between complexity and diversity? Yeah, it's a positive, not exponential, logarithmic relationship. So initially we get as we increase complexity early on, we get a really big increase in diversity, and then it tends to slow down and level off. Okay, so a couple things you need to know in general. The complexity within living organisms is hierarchical. And what we mean by that is a couple things. One is it goes all the way from like the molecular level of organization up to the organismic level, all within the same organism. Okay, so we've got molecular, and I guess if you want to start with the atomic level, you can, although that seems silly. So molecular, and then we've got organelle, cell, tissue, organ, and then I ran out of space, but then you'd go organ system, and then finally to the organism as a whole, right? These are our levels of organization, and what we mean about it being hierarchical is that as we increase a level, we're increasing the amount of complexity. I know what you're thinking. If I started higher up, I would have fit it all, and you're right. Okay, so the least complex level is the molecular level. The highest complexity is at the organismic level. It's hierarchical, and every level is more than just the sum of its parts. So an organelle is more than just a collection of molecules, right? We see that. Yeah.
What's the final level?
Organ system and then organism. They're missing. I would have to write on the wall, and people don't like that. Okay, but every level is more than just the sum of its parts. Okay, it's everything that came beneath plus organization and communication. Organization and communication. Okay, so every level is more than just the sum of its parts. Now, all animal phyla, except for one, Porifera, the sponges, they have complex tissues. So all animals besides sponges at least have this level of organization, tissue level. Sponges don't have tissue level organization. Here's that. Sponges, you could make an argument that they do have one type of tissue, but it is not a complex tissue. And so we would not grant it tissue level of organization. We grant it cellular level of organization. You're like, that's so strange, because even plants have tissues, right? And as long as they're vascular plants. So everything from ferns up, they've got vessels, they've got reproductive tissues. Even bryophytes have reproductive tissues, right? So it's just strange. We have an animal that doesn't even have this level of organization. You're right, it is strange. And that's why sponges are so interesting and so difficult to organize with other animals. Yeah?
What do you mean by division of labor?
So yeah, division of labor where you have different cells carrying out different jobs, all making up a particular tissue. So let's give the example of like muscular tissue. Muscular tissue are several different types of muscle cells, along with other types of supportive cells and progenitor cells that all function for a single task. So tissues allow for cells to communicate and divide. Oh, look at here. Here's the answer to your question. The next point. Tissues allow for cells to communicate and divide tasks to accomplish more than the cells can do alone. So it's more than just a colony, right? This is something that is truly multicellular. We've seen colonial living. We saw that with biofilms, right? We saw that with like Volvox when we were talking about just eukaryotes in general. But this is true multicellularity. Having these complex tissues that allow the cells to communicate, divide up the task, allows for really complex movements and complex sensing. And so basically, if we can get to this level or above, you expect a really highly sophisticated body plan that has a great potential for diversification, employing different strategies, living in different environments. All right, does it make sense? And this is my favorite. I'm going to show you a figure right now, and it's my favorite one from the textbook, and you'll see why. This is figure 27.2, and it's showing you, it is a representation of animal diversity. And so it chooses a black bear eating a fish, right? That's awesome. And a heartworm. I don't know if you can see this or not, but you've got this text you've got this image in your text. It's a heart just packed full of hundreds of heartworms. That's awesome, right? If you just wanted to like encapsulate animal diversity, we've got something awesome like a black bear and something even more awesome like a heartworm. All right? Any questions about that one? So again, we still see this very much logarithmic relationship between complexity and diversity. Yes? Cool. Happy Friday. All right. Our next one. So now we're going to start to talk about animal development. How have Hox genes contributed to the diversity of animals? How have Hox genes contributed to the diversity of animals? What's the Hox gene? Right? What does this even mean? Yeah, Faith.
Isn't it like, let me see if I remember, because I studied in genetics classes, isn't it like master control?
Yeah, absolutely. It's exactly what Hox genes are. Yeah, we call them master control genes. And so they don't lead directly to the production of protein, and instead they control the expression of other genes. So they really do, they work like switches, right? You may have something plugged in that is a really sophisticated machine that carries out some task, right? Like it's your phone, and it carries out the task of keeping up everybody updated and what's going on in your life, right? But then you've got a switch that controls whether or not you grant power to charge that device. And so you can have a gene that codes for a protein, but that protein never ever gets made because the master control gene shuts that gene off. This is awesome stuff. Awesome stuff. Turn off and turn on other genes, basically leading to the altered development. This is part of the explanation of how you generate all these different tissue types from cells that are all genetically identical, right? I don't know if you knew this or not, but your liver cell is genetically identical to the cells that develop your dermis and your epidermis in your skin. Genetically identical. You're like, why are they so different? And it's because they have different genes being expressed. And then we ask the question, well, why are they expressing different genes? And you answer that how? Because, what's that?
Their master genes are different?
Yeah, what did you say? Their bosses are different?
Yeah, it's like a
Oh, I like that. I like that. Yeah, because they've got different master control genes expressing or leading to the expression of some genes and not others. And so you have some Hox genes that are more active at what we would call the anterior end of an organism, the head end. And we have some that are more active in the posterior end of an organism, the tail end. We know that these Hox genes also control the presence and the arrangement of limbs and other significant figures. And we can see a lot of this has been done studying fruit flies. And so if you can get a different Hox gene to express where the wings develop, you end up developing more legs instead of wings. Which should make sense because the cells that are generating all of that are genetically identical. Or you can develop legs instead of antennae, or wings instead of antennae, right? And think about that, like a fruit fly that has wings where wings should be, but also wings where the antennae should be. How does that even work? The head is so small. You're right, it is. So small. I mean, fruit flies, the entire organism is pretty small, but. Here's where it gets interesting. You're like, Dr. Engel, this is already interesting. Here's where it gets even more interesting. Interestinger. Is vertebrates, like us, have more Hox genes than any other animals. We have more Hox genes than any other animal group. And we show significant homology, meaning our Hox genes tend to operate in the same place. So if you have the same Hox gene in a mouse as you do a human, the Hox genes operate in the same place in a mouse that they do in a human. Or the same place in a fish or a lizard that they do in a human. Yeah?
So can you use this to counter the point of embryonics when all the embryos look similar to each other?
You could use this to explain why that might be, right? Because development is not independent of programming, and we are programmed similarly. So you would expect that we would develop similarly. Is that what you're getting at?
Well, I'm saying, like, they're using embryos to prove that we're from fish.
Yes.
Can you use this Hox genes idea to say, look, we have different master control genes, so we grow differently?
You could. I don't think it would be a very compelling argument. We'll come up with lots of things that are compelling arguments of why we don't share common ancestry. That one is not one that I would die for. All right. So from this, a number of scientists will argue that what actually allowed vertebrates to evolve in the first place was a duplication in these master control genes. Right? Any of you ever run a soundboard for some application, or you've seen a soundboard, right? And so you have all these different controls of what happens. The bigger your soundboard, the higher level of control you have of what's going on, right? That makes sense? Y'all seen the movie Up? Not Up. Gosh, why did I say Up? Inside Out. Okay, Up. We could talk about that as well. I think Up. I think Up is very underestimated and is the best Pixar movie made. But anyways, that's because Up makes not everybody. Up makes 95% of people cry saying 5 words in the first 4 minutes of that movie. I don't even know what 5 words, but there's no dialogue, and 90% of people that watch it are crying. It's just powerful. Anyways, sorry. Inside Out, right? At the end of the movie, she gets a much larger board, right? Higher level of control over what's going on, which is ironic because it's when she becomes a teenager and you have less control over what's going on, right? It's so funny. Anyway, sorry. And so the idea here is if you could duplicate these master controls, then it gives you higher levels of control over the expression of all of your genes and should actually lead to higher structural complexity. Here's what's even more interesting. This is bonus information for now. It will become not bonus information in a little bit. It's like a little sneak peek of what's coming. So there's one of these Hox genes that determines this axis here, front to back. Okay, front to back axis. And in vertebrates, that gene is reversed from what you find in other animals. Which means compared to other animals, a vertebrate back is the front, and a vertebrate front is the back, because that gene is reversed. And here's what's interesting. Vertebrates, our major nerve cord is in our back. Every other animal that has a sophisticated nervous system, their major nerve cord runs along the front surface. That's awesome, right? So here's an image from your text showing a couple of things. One, the similar function of suites of Hox genes in similar portions of 2 different mammal bodies, and then 4 different complexes or suites of master control genes. And so it's a lot of genes. Again, vertebrates, we have more control genes than other animals do, by far. By far. And you know what also is cool? This is always going to be bonus information. So like if you go to an insect group like Lepidoptera, which are the butterflies and the moths, and you look at how they actually carry out their reproductive strategies, the more complex their reproductive strategy, the more Hox genes they have. Or you could say the other way around, the more Hox genes they have, the more varied their reproductive strategy. So cool, right? All right. We'll talk about what all that means, but not now. This is for later. Explain humans in terms of animal classification schemes. And I don't know, if you were asked the question, what is the most important skill to develop while reading chapter 27? You would answer it this way. The ability to classify any organism, humans or otherwise, according to these animal classification schemes. It's a good skill to develop, you know. Just in case you were ever asked that question. I mean, that seems like a weird question to ask, but just in case you were wondering, like, how do I prepare for exam 2? What types of short answer questions can I anticipate?
Can you say it again?
Sure. The ability to classify organisms according to animal classification schemes is the most important skill you can develop from chapter 27. And you're like, Dr. Engel, I don't develop any skills from these chapters. Well, make this the one you do, right? I'm sorry if that was mean. I'm sorry. I'm sorry if I hurt your feelings. I didn't mean it. I'm sure you don't think that way. Animals demonstrate 3 types of symmetry. So this is one scheme you use. Different types of symmetry. One type of symmetry is asymmetry. A means what? Without. Okay? So these are animals without symmetry. Radial symmetry. Radial symmetry is basically an infinite number of planes of symmetry. So here's radial symmetry, and it is an infinite number of planes of symmetry. No matter how you cut this on at least this one face, you generate 2 symmetrical halves. Okay? That's radial symmetry. And hopefully you're looking at that one, you're like, we don't have that, right? I don't have that. And I'm about as round as you get, right? Oh, man, that was good. That one just came to me. All right. And then the last one, bilateral symmetry. Look at this. This is us. Bilateral symmetry. Bi means what? Two. You have one plane of symmetry to cut bilaterally symmetrical organisms into symmetrical halves. Bilateral symmetry. So we have one plane of symmetry. And you're like, yeah, but Dr. Ingo, it's not perfect. You're right, our heart is displaced to the left. Faces aren't typically very symmetrical. Have you ever held a mirror up over half of your face and projected the other half? I mean, it's interesting. Anyways, and you could see, and then take a picture and do it the other way and take a picture. Anyways, sorry. It's not perfect, but we are bilaterally symmetrical. Okay, one plane of symmetry. And so organisms with bilateral symmetry, if you have a structure that occurs anywhere that's not on that plane of symmetry, the midline, you better have two of them. Or typically you're going to have two of them. You're like, yeah, but the heart's displaced to the left and we only have one. Yeah, but it starts developing in the middle and then moves. Anyways, sorry. All right, so that's one scheme based on the type of symmetry. You can also do it based on the number of germ layers. Right? What on earth is a germ layer? Symmetry, that makes sense, right? I've done that before. What is a germ layer? I'm glad you asked. So germ layers are distinct layers that appear early in development. Germ layers, distinct layers of cells that appear early in development. Distinct layers, early in development. Germ layers are distinct layers that appear early in development. So sponges, they don't have any. They don't have any germ layers. Like not even one? I mean, that's just sad. How do you have 0 germ layers? The answer is you'd be a sponge. Diploblasts have 2 germ layers. Diploblasts have 2 germ layers. And then finally, triploblasts, which are us, have 3 germ layers. Yeah, you want to know what those germ layers are, don't you?
Yeah.
Is that what you were going to ask?
No, quite.
Okay.
I was going to do, what you described germ layers as distinct layers
That develop or appear early in development.
Okay.
Yeah, distinct layers. Yeah, I'll answer that second question you had of what are these germ layers. Don't worry, we'll come to that. Okay, so that's another scheme. Here's another one. You can do it based on the number of cavities inside of the embryo during development. And I don't know if you knew this or not, but cavity is a space. Okay, we're getting some nods. Some of you knew that. Right, so if you develop a cavity in your teeth, it's because the bacteria have eaten whatever's on your teeth. They make a material called dextrin that is acidic and wears away your teeth, leaving a cavity, right? That's not what we're talking about, though. We're talking about during development. So we have a true coelom and our eucoelomates. So we have 2 cavities. Okay, and I'll show you this. We have a gut, that's one cavity. And then we have a true coelom. Coelom is a fancy way of saying cavity. So we have a true coelom, and therefore we are eucoelomates. So we can organize animals this way. Do they have a gut? Yes or no. If they have a gut, do they have a second cavity? Yes or no. If they have a second cavity, is it a true cavity? Right? What is a fake cavity? We'll get there. And then our last classification scheme. So this is for eucoelomates. Okay, so for things with a true cavity in addition to the gut, you can be one of 2 things. You can be a protostome or you can be a deuterostome. All right? If you are a protostome, your embryo is marked by spiral determinate cleavage. Like, what on earth does that mean? If you are a deuterostome, your development is marked by radial indeterminate cleavage. What does that mean? Glad you asked. We'll talk about that in just a moment. Okay? And so these are our 4 classification schemes. The symmetry, number of germ layers, number of cavities during development, and then the actual developmental progression. All right? Is it cool? All right. It is. We'll start to apply it. I mean, we made it less abstract because we use this to classify us. And so the question is, and this is our lecture break. I know it took us a while to get here, but we did our quiz first. So I want you to talk about this with those around you. Take a couple of minutes and address this question. Why do we share features with so many other animals? Why do so many other animals have bilateral symmetry like we do? Somebody give me another bilaterally symmetrical animal with one plane of symmetry. A bear. Love it. Right? And what about our other animal from that same figure? A heartworm. Oh, a fish. The fish was in that figure too. The fish was not a willing participant of the activities that were involved in that figure. Right? The fish did not sign, what is that? The consent form? So you can't reference the fish. But that's fine. Yeah, the fish also bilaterally symmetrical. That heartworm, bilaterally symmetrical. Right? Tons of animals that are bilaterally symmetrical. Anders, thank you for your enthusiasm with the bear. Thanks. So animals, various numbers of germ layers. Almost all animals are triploblast, having 3 germ layers that develop early in development. Why is that? Glad you asked. You can talk about that with those around you. Different number of cavities. Almost all animals are true coelomates, like we are, that in addition to the gut, have another cavity that's a true cavity. And then protostomes and deuterostomes. This is one where we're not in the majority. The majority of described species are actually protostomes, and we are deuterostomes. I didn't even tell you that. Man, I didn't even put that one on here. We are deuterostomes. The majority of described animal species are protostomes. This is one where we're not in the majority, but there's still a whole bunch. It's a technical term. And it's all together, a whole bunch. So the question is, why do we share so many features with so many other animal groups? All right, 2 minutes. Starting now.
It is true. It is true. Done. We answered it. Why do you?
Even though the relationship might be different, but I guess with the ability
You had 7 in your group? Did you put Allison on yours? Did you get them all right? Yeah, I'll still give her the option if she wants to do it. I'll just remember. Did you all put 8? Did you get them all right?
You have the tail.
How handy that would be? Yeah, exactly.
30 seconds!
Turn on the light.
Oh. Turns into ace. That's the weakness right there. Wait, so he turns into monkey? Or like come out? Yeah. That's his curse? Oh, so that's why they just
All right. Somebody explain this to me. Anders.
I think we have the same characteristics because we need the same things to survive. We reproduce the same way.
Yeah, so you have to, so you're noting, let me see if this is what you're saying. We function in similar ways to other animals because we have similar needs. Function is not independent of structure. Therefore, if we function similar to other animals because we have similar needs, we should also be structured similar to other animals. Is that what you're saying? I love it. almost the same amount of enthusiasm as the bear. It's good. I love it. Yeah, if you're going to answer, I mean, commit to it, right? Sell out. What do we got? Some other ideas. Chloe.
I think you can like look at it depending on like your perspective. If you look at it as a biblical or evolutionary, we could have evolved from.
Yeah, absolutely.
Similar.
Yeah, I mean, you can't make an argument that that is not a reasonable conclusion, right, to draw. That the reason why we are structured similarly is because we share common ancestry, right? You can't really argue that that is not a reasonable conclusion. You could argue whether or not that conclusion is possible, right? It's a different story altogether. So let me just give you a real quick example of this. So you have what's called the Cambrian explosion. We'll talk about this in a little bit. We won't get to it today, but we'll probably get to it on Monday, where you have basically all of your animal phyla that show up all at the same time with no transitional forms of how you got there from one original animal. You cannot argue that the best explanation of that is anything other than that these are all separate creations that were present at the time that that fossil record started. I mean, you can't argue that that is not the best explanation. But if your philosophical commitments don't allow that explanation, then you don't have to consider it, even though it's the best explanation. In the same way, the best explanation, and I think I did this already with Emerson and I, right? With common ancestry, where like Emerson shares more genetic markers with her siblings than she does with me because they have a more recent common ancestor, right? And then you just say, well, the reason we share more genetic markers with chimpanzees than we do with any other animals is because we have a more recent common ancestor. You can't argue that that is not the best explanation. But if your philosophical commitments don't allow that to be a potential explanation, then you don't have to deal with it, right? It's wonderful. Now, we have alternative ways to explain that in the same way that if you're committed to methodological naturalism, you could explain the Cambrian explosion other than that they were all separate creations that were present at the time that the fossil record began, right? There are alternative explanations, some of which are good, some of which are not so good, and we'll ignore the not so good explanations, but we'll deal with some good explanations. All right? Make sense? What are some other reasons? Anyone else want to venture some reasons? So Anders gives us that we have similar needs, right? All living organisms have to carry out the characteristics of life. Here's an interesting rebuttal to that argument. Not to say that I don't love it. I love it. But here's an interesting rebuttal. We have the same basic environmental necessities that insects do, and yet they are protostomes, we are deuterostomes. Right? Like it's like we've got to metabolize and make copies of ourselves and respond to stimuli and maintain homeostasis. And that one seems to be somewhat arbitrary how you develop early on. Although I will grant you, Anders, that you can't get identical twins in insects like you can in humans because they're protostomes. You're right about that. Yeah. All right. Nobody else has any explanations? Nobody wants to go out? All right. So here is basically the first couple of stages of development regardless of the animal. Okay? So first stage is zygote. This is after fertilization. Now you have restored the diploid state, right? Germ cells start as diploid and then they develop into either sperm or ova and they are haploid. After fertilization, they are now diploid. We've restored the diploid state. We've got zygote. From there, we get to an 8-cell stage. From there, we get to the blastula, which is basically a hollow core of cells. And then from there, if we have some germ layers, we start to develop distinct layers. Here, you just have one ring of cells, no distinct layers. Now we've got 2 layers, an inner layer and an outer layer. We've got a diploblastic organism. And then here's based on symmetry. Here are sponges with their asymmetrical body plans. Here is an anemone and a jelly with their radial symmetric body plans, right? No matter where you cut this on this plane, you have an infinite number of planes of symmetry, radial symmetry. This is cool. It allows an organism to interact with its environment without moving in 360 degrees. All right? It's great. Same thing with our jelly. And then here's a butterfly, a lepidopteran, with bilateral symmetry. Okay? Animals with bilateral symmetry usually have a head. Head and bilateral symmetry tend to go hand in hand. You're like, what about if I pulled the head right off of that butterfly? Is it still bilateral? Sorry, that's something my son would ask. Yeah, they do still have bilateral symmetry. But then some organisms have bilateral symmetry and lost the head, like tapeworms. Tapeworms don't have a head, and they are still bilaterally symmetrical. You're like, what is that creepy looking thing at the anterior end of a tapeworm if it isn't a head? We'll get there, don't you worry. We'll talk all about tapeworms and you'll love it. So here is a bilaterally symmetrical organism. Here is the sagittal plane, which is our plane of symmetry. And then we have our transverse plane that cuts us into anterior and posterior halves. And then we have our frontal plane that cuts us into, in our case, since we're upright walking, anterior and posterior. But for most animals would be ventral, dorsal. But since we're upright walking and we use the term anterior to be the direction of movement, then we have to, adopt that. So then you're like, well, then what do we use here? I'm glad you asked. We use superior and inferior instead of, and don't worry, we'll come back to this and you'll love it. That's basically every one of our dissections, it's going to reference one of these planes and it's going to reference like a corresponding section. You're going to have to know how to handle it so you'll get comfortable with those terms. Don't you worry. Don't you worry. Here's germ layers. Here's a diploblast. So we've got a layer of cells here in the ectoderm. We've got a layer of cells here in the endoderm, and then it's filled with this non-living in a jelly. It's jelly. It's like this really weird viscous material. Yeah. I read an article, I don't know, maybe 2 weeks ago, that said at the rate we're going, the oceans in 100 years will be filled with nothing but jellies because of overfishing and other things. Anyways, that was kind of fun. So I started picturing an ocean full of jellies and then thought it'd be pretty cool. If you got enough jellies there, you could run along the surface and never get wet because you're just bouncing on top of jellies. Oh man, that'd be fun. All right. And then here's a triploblastic organism. You've got ectoderm on the outside, endoderm on the inside, but now we have a living cellular layer on the inside, the mesoderm. All right. Oh, here's the cavity, right? What does a fake cavity look like? So here's the digestive. So this is animals that at least have a gut. Okay, because some animals they don't even have a gut. We don't even worry about naming cavities here. Okay, so here's the gut. There's no other cavity. So we call them acoelomate, without a coelom, a gut or a cavity in addition to the gut, right? So clever. And then the eucoelomate, like us, here's this cavity in addition to the gut. We've got a cavity in addition to the gut. We are a coelomate, and we're a true coelomate. And then here we have a pseudo-coelomate, which means fake cavity. You're like, how do you have a pseudo-coelom? That doesn't even make sense. And basically, it's not a fake cavity. It's just not lined by mesoderm on either side. See, in the eucoelomate, there's mesoderm on either side, mesoderm on the inside, mesoderm on the outside, mesoderm on the inside, mesoderm on the outside, right? Mesoderm on the inside, mesoderm on the outside. In the pseudocoelomate, the mesoderm is only on the outside. It's not on the inside. You're like, what are some of the implications of that? We'll get there, okay? But if I told you this, that muscles develop from mesoderm, do you see anything that might be difficult for these animals? See if anybody can pick this up. If you can't, it's fine. We'll get there. I'll give it to you. Tara. Yeah, what about digestion? You have no muscles lining your digestive tract. That's really interesting. I don't know if you knew this or not, but most of the materials move through our intestines by alternating muscle contractions called peristalsis. And if you actually, those muscles can become paralyzed and material can get stuck in your intestines. So animals without that muscle lining of their digestive tract, the only way they can keep material moving is to keep packing material in. It's like you just got to keep pushing it in order to get it to move through. Or you put an enormous amount of pressure on your digestive tract from this cavity here full of fluid. And this is pretty cool. I don't know if I'll get another chance to tell you this. So there are some animals that the pressure inside this cavity is so high that if you open up the anus, it'll shoot feces like 400 times the length of the animal. It's awesome. You're like, who did this kind of experiment where those heartworms are one of the animals that are like this. You're like, who thought, let's take a heartworm and let's open up the anus and figure out how far it can shoot its feces. I mean, it's such a wonderful, I don't know why I didn't think of it. Yeah.
They don't have mesoderm on the inside, would they not have internal organs either?
They can, but they're going to be limited in terms of the sophistication of their gut. So so they still have mesoderm here, and you can develop some of your internal organs outside, but it's going to simplify the gut, right? A lot of our internal organs, like stomach, intestines, small and large, you find hearts in these organisms. They just develop out here and away from the inside of the organism. Yeah, but it's a very simple. I mean, it's just basically a tube. And really, considering what happens when you open up the anus, it's really just a cannon, right? All right, have a wonderful weekend. Enjoy the rest of your Friday.